Next speaker is Max Fuxjager from Wake Forest.
Hi. Yeah, I'm Matt Fuxjager. I'm from Wake Forest. So I'm a behavioral endocrinologist, and I postdoc with Barney, and I've recently started at Wake. And so I'm interested in how evolutionary forces influence hormone systems to drive kind of species variation in behavior, potentially explain some of this variation in behavior we see across species. And so this led me to work in mannequins. I started off working in mice. Given that they're, affected strongly by sexual selection, and we know they have an androgen-dependent behavior, these displays. And so I've started, in Barney's lab, we started looking, and I've kind of pursued as well, looking at sensitivity of the neuromuscular system or the neuromotor systems to androgens, right? So we find that in the golden-colored mannequin, the neuromotor system is highly androgen sensitive. And if you block androgen action in these areas of the neuromotor system, you block display performance. And so what we've gone on to show is that you can, through work with Laney and whatnot, you can quantify the complexity of a lot of these different species displays, and we find a nice correlation between display complexity and androgen sensitivity of the neuromuscular system. And so this, of course, has led us to think about, well, what are androgens doing to these tissues? And so we've worked a lot in the muscle, or I've worked a lot in the muscle, the skeletal muscle of the wings, to see what androgens are actually doing to influence the tissue so that they can facilitate a lot of these crazy display moves, some of which are more complicated or they require faster movement than others. And so we've used RNA-seq to get at genetic candidate genes, or candidate genes that might underlie muscle performance. And then I've also started getting to a lot of physiological measures to assess what are these muscles capable of and how are androgens actually influencing what these muscles can do. So that's it.